This is the second half of the second half of fermentation of carbohydrates. What I'm doing is I'm separating these into two separate videos because the MRVP test, it says to allow the bacteria to inoculate for 48 hours or more. And it hasn't been quite 48 hours, so I'm going to let them go longer. So I'm going to do this part of it first, and then there'll be a second video with the MRVP demonstration. Okay? So let's go ahead back to the first part. Now this is where I had assigned you a particular bacteria that I wanted you to do. One group did E. coli, one group did Enterobacter, another group did Pseudomonas, and a third group did Proteus vulgaris. So what you'll have to do is share your results. What I'd like for you to do is look on page 97. In the data table, it says it has the 4 bacteria on the left, and then it has 24 hours and 48 hours. Well, obviously we're not looking at them at 24 hours because it's already been almost 48. So what you'll do is you'll just draw a line through the 24-hour. If we met every day, that's something that we could do. But unless you wanted to come in on Thursday to look at them, we won't be able to do 24. Now, I am taking them out of the incubator at 48 hours. So that particular one, what you'll see is when I took them out of the incubator at 48, even though you're looking at them a week later, okay? In that table, it has the glucose, lactose, and sucrose. So you had put your bacteria into the 3 separate fermentation tubes. And then you'll fill that out, and then you'll go to another table that had a different bacteria or another group and fill out their information. Now, we had enough for 8 groups. So we had 2 groups doing each type of bacteria. What I want to make sure that you do is not only do you look at your results of your bacteria, but also look at the results of the other group that had the same bacteria and make sure they match. Because somebody may have made a mistake, I won't name anybody, no, just kidding. It's possible that somebody could have not inoculated it properly and you're going to get different results. The other thing is if you tightened your tubes on too much, you're not going to get a result, okay? So make sure that you match with the other one, or sometimes you don't get enough inoculum in, enough bacteria, but maybe the other group did. So if you didn't get results, look at somebody else, but definitely compare what you did to what other people did. The other thing is you'll have to go and find the other bacteria, the other cultures that were done. So to fill out that table, you're going to be required to go around the entire classroom, look at their tubes, interpret the results, and work with the other groups, okay? So to fill this out, the first thing you're going to look at is growth. So what did I say growth was? Well, I said it was turbid, okay? So if you see some sort of cloudiness, that means there was growth. Right here is an uninoculated tube. It's very clear there's no growth in it. I pick up one of these, I can definitely see the cloudiness. So there's definitely growth. I don't particularly see any that did not, well, this one looks like it might not have grown. So give it a little tap. It's slightly cloudy, so there's very little growth. The reason that you have to check for growth first is because I can't interpret the results as being positive or negative until I make sure there's growth first, okay? If I see something like this and I say, oh, it's negative for lactose fermentation, well, that's wrong because actually it was no growth. So I have no determining factor of whether or not that bacteria is positive or negative. Now this one does have a little bit of growth in it, so I could say, yes, there was growth, okay? Now, the next column says the color. So was the color red or yellow or pink? Some of them turned pink, okay, and that means something. So remember, this is uninoculated. So they started out with a reddish color, okay? If they stayed red, that means that they did not ferment the sugar. Because remember, the phenol red turns yellow in the presence of acid. And acid means that they fermented that sugar. So the color would be red, yellow, or pink, okay? Acid, yes or no. If it turned yellow, yes, that's acid. If it stayed red, that means no acid, okay? The last thing was gas. Let's go back to the acid. If it turns pink, that means that it used up all of the sugar and it actually started using peptone, okay? So it's not really, it's more of a it's not acidic, it's actually basic. You'll get that hot pink color. Okay, now looking at gas. Now remember I told you to guys look inside the Durham tube and detect gas if there was a gas bubble the first time? This one right here, it's hard to tell. There's a little gas bubble in the top. So I would, if I was this person, I would go and look at my note and see if there was gas to begin with. But I can definitely see a gas bubble there. This one right here has a huge gas bubble. Okay, it's about that big. So this one is definitely positive for gas as well as acid. You may have some that produce gas but not acid, so you'll just have to take a look at those. So then you'd fill out that entire table and then interpret the results. What does it mean? Okay. Now the next table is going to be the MRVP, which I said we were going to do on another video. Okay, so for this part of the fermentation of carbohydrates, that was all you needed to do. There were two other things that you did. You inoculated that plate with a McConkey agar, and I'm still not sure what we're doing with those plates, so just for now, don't worry about them. When we get to class, we'll talk about them. The other one was I asked you to inoculate the Simmons citrate tube. So this was the green tube. And if you refer back to page 107, you learned that the Simmons citrate will turn blue. Hope you can tell the difference. Will turn blue if that bacteria uses citrate as its sole, citric acid as its sole source of carbon. So this one is a positive for citrate. And I had told you that I wanted you to do that extra test. So on your table, you could just add an extra column with the bacteria to say if it was citrate positive or negative. Okay? And I just added that on because a previous edition of the book had it on there. And like I said, later in the semester, you're going to be doing a test with citrate. So now you know what it is and it's not something new to you.